we're also delighted today to welcome back Dr. Sophia Vinogradov. I'm having trouble speaking this morning. She is, as you know, our distinguished visiting lecturer speaker this morning. She's the Donald W. Hastings Endowed Chair in Psychiatry and head of the Department of Psychiatry and Behavioral Sciences at University of Minnesota Medical School. As I think many of you know, are old friends, but she received her undergraduate degree from Yale. Then she did her MD from at Wayne State, and then her psychiatry residence down the way at Stanford, residency down the way at Stanford, where she also completed a psychiatric neurosciences research fellowship at Stanford and the Palo Alto VA. Jesus, can someone get me a cup of coffee? Palo Alto VA Medical Center. Dr. Vinogradov has been a pioneer in clinical translational neuroscience, focusing on cognitive dysfunction and psychotic disorder. And I saw, interestingly, on your recent grant, also looking at autism spectrum disorders. So I love that. She's been at the forefront of computer-assisted brain training to address processing abnormalities associated with these conditions. Her laboratory uses a range of methodologies, MEG, EEG, fMRI, to probe brain changes in both early sensory processing and higher order cognitive operations in people, particularly who are undergoing cognitive training. Her work is long focused on young adults in early phases of psychosis with the goal of providing intensive early interventions to ensure optimal outcomes. Sophia is a prolific author, total of more than 190 peer-reviewed publications, more than 20,000 sites. She's currently the PI on 6 active grants, including a P50 and R01 from NIH, which, given I well know the responsibilities of being chair and all the other things that you do, and that last bit is really a remarkable track record. Despite her many responsibilities as department head and as a lab director, she also leads a clinical neuroscience curriculum for the residency training program in her department. She's been the recipient, not surprisingly, of multiple awards, including the NAMI NAMI's Exemplary Scientist Award, the NAMI's National Scientific Research Award, the Dean's Distinguished Research Lecturer Award at UMN, and the Alexander Gralnick Award for Research in Schizophrenia from the American Psychiatric Foundation. And of course, Dr. Vinogradov is UCSF family. As I believe you all know, she was previously our vice chair at the San Francisco VA Medical Center, as well as associate chief of staff for mental health. It's wonderful to have you back. We had her here in 2022. We are praying to the gods of IT that this goes better than that last time. Today's talk will address the role of neurocomputational profiling in parsing heterogeneity in psychosis. Dr. Vinogradov, it's lovely to see you. Welcome back.
Thank you. Good morning, everyone. It's such a joy to be here. And I feel like I'm at this sort of wonderful phase of my career where I get to tell you how work that I really began with my career here at UCSF has continued to flower.
It is on. Oh, okay, it is working. Yeah, I'll just
generate excitement all over. You know, again, if it weren't for my roots here, I wouldn't be able to tell you about the work that I've currently embarked on. So this is a long and complicated title, but in some ways, if you take home nothing, no other message, the message is, and I'm doing this to be provocative a bit, is that really the way we've been thinking about psychosis spectrum illnesses hasn't been working very well. And there is starting to be a paradigm shift in how we even just conceptualize these illnesses through the use of essentially mathematical modeling of aspects of behavior. And this is an approach which is really not just within psychosis spectrum illnesses, but we're starting to see pervade other ways of thinking about psychiatric illnesses. And it's my hunch that this is actually where our field is headed. I'm not the only one to be thinking this, of course. And so if you have no expertise in this area, I want to just start to give you a taste of the frameworks that are used and why those of us who are beginning to do work in this area think these frameworks are going to be useful for drilling down into the incredible complexity of what the brain does or doesn't do when it malfunctions to create psychiatric illnesses. Here are my disclosures. Really, the projects I'm going to be talking to you today, discussing with you today, They're funded by NIMH, and they're not related to some of these other funding sources. So I'm going to be discussing principles and ideas with you which emerge from the interactions of this wonderful team at the University of Minnesota, and I hope I can communicate throughout that these are ideas that have been part of a group effort. They're synergistic ideas are coming from all these wonderful people. I won't name them all for you. Those of you who do work in this field will recognize some colleagues who are of course quite well known. I do want to highlight David Reddish here. I'm learning how to use this new pointer with his spotlight, and it's kind of fun, actually. David is my co-facilitator in this work. He's a computational neuroscientist, and we've put our heads together and really have embarked on this journey with these wonderful people. So there are three highlights that I'd want to emphasize with you today. The first is this idea that there's an elephant in the room, at least in terms of psychosis research, and that there's a lot of parts to the elephant. My second point is that some of these methods, which have been moving into psychiatry from the field of computational neuroscience, it can be a helpful linking tool for understanding all these different parts to the elephant. And then I will conclude with a very brief and probably for some of you a little bit overwhelming tour of what we might discover when we use this kind of a linking tool. But again, what's going to be important is not so much the details of what we're digging into as the principles of how we're applying them and how they might eventually be useful for both understanding illnesses and thinking about tailoring treatments to very specific aspects of an illness. So what do I mean when there's an elephant in the room? Well, again, I want to be a bit provocative, but it's the idea that the way we've been thinking about psychosis spectrum illnesses, and you're going to hear me use the term psychosis and psychosis spectrum illnesses sort of interchangeably, and I'm really talking about these group of disorders which are characterized quite fundamentally by the fact that a person is vulnerable to slipping in and out of episodes of psychosis or remaining in them for long periods of time. So it's not been working very well. We've got 50 years of research, some of it really incredible research, but we haven't had really significant breakthroughs. Again, I'm being a bit provocative. As someone who also practices clinically, I would say probably the most significant breakthrough has been the idea that, you know, the earlier we identify and intervene, probably the better we are doing by our patients. But the early interventions that we are offering right now are not really based in a fundamental, profound understanding at the neuroscience level of the actual pathophysiology that's happening in the disorder. The treatments we're using, even in early interventions, which are showing promise, are behavioral treatments. There are things like CBT for psychosis. There are things like individual resilience therapy, family therapy. There are these behavioral psychosocial interventions. They work if we can get people to deliver them, right? There's workforce shortages. If we can get patients to engage them, because there's motivation and cognitive difficulties that are impeding people's ability to engage. So we have things, we're doing them. We've had a conceptual breakthrough that it's important to intervene early in the course of illness, but the tools to really accelerate and change outcomes in terms of helping this brain that is not functioning very well function better, we don't really have those yet. So why might this be? Why have we had these 50 years of research but no real breakthroughs? Well, here I wanted to highlight a study that Judy Ford authored not so long ago. And what Judy did, again, working with a team, is take this large imaging data set. And what she looked at is what's the relationship between, well, what's different about connectivity relationships in the brain when you look at people who have schizophrenia who have visual hallucinations versus those who don't. Now, what you see is in green are the visual hallucinations. And of course, I don't have my glasses on, so I'm not going to be able to tell you what the other ones. I think red and blue are with and without auditory hallucinations and no hallucinations. So these are all people with schizophrenia. They're showing different symptoms. And the point here is that each graph is showing a connectivity pattern between the amygdala and other brain regions. And again, the point being that people who have visual hallucinations have these different connectivity patterns from those who don't. So that's interesting, but it's already telling you that there's some kind of heterogeneity in the illness, right? Different connectivity patterns are showing up in different patients. depending on what their symptom manifestations are. Well, you'd think to yourself, well, maybe this is a great breakthrough, and maybe this is going to let us drill down into something really fundamental about this particular subgroup of patients. But in fact, it's turned out to be more complicated than that. Partly because when you think about what are visual hallucinations, and this is a review done by a researcher named Steve Silverstein, who thinks a lot about the visual system and schizophrenia, there's lots of ways to think about what visual hallucinations are. They can be at this very simple distortion level. They can be a kind of simple hallucinations. They can be more complex and elaborate ones. And there seem to be many different pathophysiologic factors in the brain that could be giving rise to these various kinds of, abnormal visual phenomena. And so you've got this heterogeneity again in the underlying pathophysiology and in the causative factors that might be combining in different ways in different patients to give rise to this phenomenon that we then end up observing clinically. So in a sense, there's probably many reasons for the lack of breakthrough. One of them is like, how do we measure the things that we think are important clinically? Like we say, okay, this person has visual hallucinations or auditory hallucinations, but how do you really measure that, right? It's a subjective experience. That's sort of one reason. There's a lot of variance in the constructs. Anyway, those are almost kind of, if you will, important aspects obviously that we need to think about and tackle, but there's also this idea that maybe we're looking under the wrong street lamp. In other words, overt psychopathology, the symptoms we see that patients manifest, and this is going to be not just in psychosis, but in other illnesses as well, it's likely a final manifestation of multiple latent processes that are happening in the brain and different pathophysiologic contributors that are happening in the brain, some of which may be compensatory. I think that's something we also forget about in psychiatry. As the brain is developing, perhaps as it's developing with some aberrant, say, neural microcircuit kind of functioning or aberrant macrocircuit dynamics, the brain is going to compensate aspects, right? And we have evidence for that. But we don't often take into account these models of compensatory processes. And then finally, something which is near and dear to my heart is this idea that, we focused a lot in the last 50 years on the positive symptoms of psychosis, hallucinations, delusions, thought disorder, and so forth. But actually, what really impedes people's functioning and their ability to have a kind of satisfying life is the impaired cognition that is so fundamental when people have these illnesses, and the impaired motivation. We now have a wealth of evidence showing that both of those domains, motivational systems in the brain, cognitive systems in the brain, are very disrupted. And these are the things that determine people's actual psychosocial functioning. But they're not so easy always to measure well. So a bottom line that I want you to be thinking about as we move forward now, and this is a nice summary of this really interesting kind of multi-site project called BSNP. Some of you may have heard of it, which is bipolar disorder and schizophrenia network intermediate phenotypes. And I'm just going to walk you briefly through what BSNP did because it again sort of captures, it's a really interesting data set, and it captures some of what I've been discussing, which is what they did is they recruited patients from who had three different kinds of diagnoses. They had either schizophrenia, schizoaffective disorder, or bipolar disorder. And then all of these patients got a range of different biomarkers. They were called a psychosis group. They all had psychosis. And then they went through a bunch of different biomarkers, electrophysiology, brain structural imaging, cognitive measures. They created a global neurobehavioral cognition score, symptom measure, various kinds of symptom measures, functioning measures, and so forth. And then they applied some machine learning cluster analytic techniques. So what that means is in a kind of atheoretic way, they sort of queried all of these physiologic measures they took, and they said, in a sense, data set, tell me what groupings exist within you. We're going to forget about diagnosis, just looking at the biology. And what they saw is that three different groups or clusters of these different measures emerged, and they ended up calling them biotype 1, 2, and 3. And these three different clusters all carried people who had any of the previous diagnoses. So again, the idea here is that you had people who had schizophrenia, schizoaffective, and bipolar illness in biotype 1, similarly in biotype 2, biotype 3. And what grouped these people together was not their DSM diagnosis, but their physiology. And so biotype 1 had poor cognition, it had more brain atrophy, it had lowered electrophysiologic responsivity in the brain. So it was like a hypoactive brain in some ways. Biotype 2 was more of a hyperactive, hyperexcitable cortex, fewer cortical structural findings, more subcortical findings. And then biotype 3 was sort of intermediate between what was seen in normals and what was seen in the impaired group. So what's interesting is that these biotypes emerged, and in terms of where this research group is taking this work, it's still kind of hard to make sense of what it means. Like, they're grouped, okay, DSM diagnosis doesn't match to each group, but now what? This is sort of where we're at. So the way we like to conceptualize this, and we're not alone in this, but this comes out of work that Angus MacDonald put together with David Reddish, is this idea of equifinality and multifinality. And again, this is not going to be true just in psychosis spectrum illnesses, but in other psychiatric illnesses as well. So it's this idea that you have multiple causes, right, that can lead to the same outcomes, in this case, symptoms A or symptoms B, and multiple outcomes that can arise from similar causes. And so this is just the idea that in probably any psychiatric illness, certainly in psychosis, we know that there's going to be a range of genes that are going to contribute. These are going to interact with different environmental factors. That's going to create cellular abnormalities, which is going to lead to different kinds of cognitive or affective processing abnormalities and so on. I think you get the picture here. I would say, interestingly, this diagram, what was sort of left out, and I want to insert it because it's an important level of analysis as we try to make the tie between different causes and outcomes in a psychiatric group of psychiatric illnesses, is the microcircuit and macrocircuit level of analysis, right? A gene defect or a group of gene defects, or not even defects, just variants that combine with environmental insults, they're going to affect microcircuit functioning. That's going to translate into large-scale macrocircuit dynamics in the brain, which of course is eventually going to affect cognition, ability to process information in the world, and give rise to then to the illness that we see, those symptoms that are manifested. So this implies that there's a lot of parts to the elephant. And in fact, those of you who are in the field of either treating people with psychosis or doing research in the area know that there's a lot of different models and there's a lot of different areas of inquiry out there. And I'm just going to touch upon them again, not doing justice to any of them, but just to illustrate all the different ways people are looking at the elephant. And it's a bit daunting, and I've probably left a few out, but some of them are, well, there's excitation and inhibition imbalances that are happening in the cortex, as well as hyperdopaminergia that's happening subcortically. And either alone or in some combination, this is giving rise to some of these microcircuit and macrocircuit aberrancies. So that's one model. You've heard maybe about the glutamate hypothesis, that there's a reduced glutamatergic functioning in cortex. You certainly have all, if you've ever treated a patient, have heard about hyperdopaminergia because we give dopamine blocking agents to treat patients and so forth. Then there's the prediction errors and aberrant salience models of psychosis. And again, not to dwell on it, but this is the idea that abnormalities in information processing in the brain are leading to changes, aberrancies in how the brain is deciding what's salient in the environment and what isn't. And the brain is over assigning salience to random stimuli and then trying to create a narrative around it. And it's doing that because it's not very good at making the right kinds of, or experiencing the right kinds of prediction errors. In other words, it's not able to create a model of the world in such a way that when that model gets disrupted by something, either truly salient or aberrant happening. It can say, oh, that's truly aberrant. That's salient, has to be classified in a different way. It's again got this aberrant assignment of what's salient and what is to be predicted as happening in the world versus what is not kind of predictable in the world. And then there's some ideas about which circuits are involved there. There's the inflammation and immune dysregulation model. There's been a lot of exciting work in this area in terms of microglial overactivation, over pruning of synapses in psychosis, again, with ideas and models about many different factors that might be leading to that etiologically, both peripheral inflammation, central neuroinflammation, primary dysfunction in complement, which is leading to sort of these microglial abnormalities. And then, of course, all sorts of symptoms being explained. There's ideas about disrupted neuroplasticity mechanisms, that genes that are very active and involved in how the brain is able to sort of remodel itself and learn in response to the world, that again, there's disruptions there. And what the final endpoint is that there are maladaptive plasticity mechanisms occurring, altered plasticity mechanisms, either too little or too much happening. And then finally, but not finally, because again, I'm sure I've left out a few, is this a notion of what's called the triple network model in psychosis. It's the idea that there are aberrant dynamics linking three important networks in the brain. And these three important networks are the salience network, which is really responsible for detecting and integrating information, task relevant, important information in the environment, and helping the brain toggle back and forth or switch back and forth between the default mode network, which is when the brain goes into more offline, self-referential kind of thinking and processing, versus the central executive network, which is where the brain switches on and starts to do sort of task relevant problem solving, actually, making decisions to move one appropriately through the environment. And so there's this notion that there's some kind of altered dynamic so that the brain isn't switching back and forth properly between this more internal self-referential state, which again, you could start to see, oh, that might explain some of the symptoms we see in psychosis, and the ability to problem solve, be online, do task relevant operations. So this is complicated, right? How could you ever put all the parts of this elephant together? The multiple causative factors, they're obviously going to be interacting in complicated ways. And basically, as my colleague David Reddish likes to say, the journey from neuroscience to psychiatry is long and complicated, right? And none of us would disagree with that. But the premise that our group has been working from is that the tools of computational neuroscience can be a helpful linking method to link across these levels of scale. Like, how do you go from what might be happening, what might be altered at a cellular level, to then what's happening at a microcircuit level, at a macrocircuit level, finally emerging into kind of the behaviors or the group of illnesses. So this is based on the premise that the brain's an information processing organ, i.e. a computational organ. We know that when we change the brain through giving drugs, through injury, through genetic variants, we change mental processes. I have always been interested in interventions, behavioral, cognitive training interventions. So it's also important to point out that changing mental processes changes the brain also. And in fact, as I've mentioned, in schizophrenia, particularly early phase of illness, we know that if we can get patients to change some of their mental processes, then we are likely, and in some of the work we've done with cognitive training, have evidence to show that we're changing how neural systems in the brain are interacting with each other. The problem is that in psychosis, likely because of some of the inherent limitations that are created in really fundamental aspects of information processing in the brain due to the genetic and molecular and environmental effects on the brain, those changes from behavioral interventions are slow and effortful and take time. And so the idea would be, how could we accelerate them? How could we create a combination of more fundamental neuroscience-informed interventions that helps the brain be prepared or have more in a sense, improved ability to make use of these behavioral kinds of interventions. You've heard me use the term computational psychiatry. I just want to clarify for those of you who don't do work in this area that in some ways it's important to think that there are two kinds of computational psychiatry methods. The first one are these machine learning methods that can be used to characterize measured features in an illness or group of illnesses in an unsupervised manner and give you new insights. That's like what the BSNP consortium did, where they took all these biological measures, kind of mixed them in a pot, and then did machine learning methods to separate them out again. But the approach we are focused on, and which has been kind of really kind of put forth by Josh Gordon, who is the director of the NIMH, though soon moving to another role, and in a book that he and David Reddish co-edited, is the idea that the brain carries out computations. Changes in computations produce psychiatric vulnerabilities, and we need to understand that if we're going to identify the vulnerabilities and think about new treatments. And there are two kinds of computational modeling that can be done kind of under this rubric. And one is focused on computational modeling of how neurons might actually be interacting, or microcircuits in the brain might actually be interacting and processing information. And the other is focused on taking behaviors, dissecting out behaviors, and then focusing on developing models of how really at a trial by trial level, as an organism moves through a behavior, what is the information flow that is occurring? And we've used both methods, and I'm going to be talking more today about that information flow kind of model generation, as opposed to the more neurophysiologic-based models, although people in our group are working on those as well. So the idea here is that computational psychiatry methods, when successful, can be a linking tool that you can then use across species. And of course, working with different species allows you to do different levels of investigation and therefore different levels of scale. So if you can come up with a computational method where you can have a mouse, for example, do a certain task that has close analogies to what a brain might do if it were a human brain or a monkey brain, then you can do things in the mouse model to see how computations are either enhanced or disrupted that you couldn't do in the human model, but that could give you insights to what similar disruptions that you might observe in humans are due to. So you can do genetic manipulations, you can do gene editing, for example, you can do slice electrophysiology and so forth. Similarly, if you can do something similar in a macaque, in a monkey, again, you can do the kinds of recordings, whether it's single cell or dense recording across neuronal ensembles, that could give you insights that you could never get from being able, because you can't do those experiments in humans. So the idea here is that, again, mental processes that we see in psychiatric illnesses are emergent phenomenon, just like a traffic jam might be an emergent phenomenon, or traffic that's flowing very well in a complex freeway system. And you know, the notion is you can't predict traffic from the nuts and bolts of a car, and that's sort of the idea of, you know, neuroscience can tell us about nuts and bolts, but it's not going to be able to tell us on its own very much about you know, the traffic patterns. But you can explain traffic patterns if you know the physical properties of cars, the arrangements of roads, you know, timing of traffic lights, psychology of the drivers, time of day, et cetera. That, and provided you have a set of tools or methods that allow you to create these links across these levels of scale, and then to understand the interactions across the levels. So that's the hope. So this fundamental idea, which I think has been implicit in what I've been saying all along, is that the brain processes information, carries out computations in order to support our adaptive functioning in our environment. That's what it evolved to do. That's the whole purpose of it. And as I think is self-evident to people in this audience, you know, you've got kind of genes and molecules and cells, and they're assembling into microcircuits and then into macrocircuit dynamics, which of course is what allowing the brain to do all of its multiple computations and then leads to behavior. And here are some of the different kinds of information processing groupings, right, that the brain has to do. This is just a few of them, but they happen to be the ones that are most important for us for the higher order things we're interested in. So the brain has to have computations for perceiving information. It has to have computations to create context, recognizing the environmental context. And then it has to be able to maintain that environmental context correctly. So you carry out your right behaviors. It has to have computations that allow it to engage in motivated behavior and learning. In other words, assigning value to what it's doing in the world or to what the world is presenting to it, so that it can, right, again, learn and adapt to the environment and do the correct action outputs. So again, I wanted to say this, the brain is a bidirectional information processing organ. And we know that if we can change how a person is functioning, or even how they're thinking about their world, or even how they're processing information in the world, then we can change some of the computations in the brain. And likely, although this hasn't yet been proven, we've certainly shown it at the macro circuit level in humans, but likely have this downstream effect as well. So what I've learned from working with computational neuroscientists is they like to think about these processes that the brain does to represent the state of the world adaptively as having three major components to it. And this is the language I'm going to use moving forward here. So the brain's trying to set up these accurate state representations, and they have to be stable. So And what does it need to do? It needs to estimate the state of the world, state estimation processes. It needs to maintain the stable representation of the state of the environment currently. And then it needs to engage in state learning, and then, of course, generate state responding. So these are the three ways that my computational colleagues think or group some of these processes. And we're going to talk about them just a little bit more. And in a sense, the core tenet of our P50 Conti Center, which is a program project funded by NIMH, is that psychosis arises from disruptions in how neural systems process and represent information about the state of the world, state representation. And so if we're going to really discover new ideas about new treatment approaches, we need to understand this computational processing, the state representation processes. What we need to do is understand how they differ between people who have vulnerabilities to psychosis and who do not. But even more importantly is how it might vary within the group of individuals who have psychosis experiences. Again, our core tenet being that, okay, if you've got these, say, three major areas that can be disrupted in terms of how the brain is representing the state of the world, it might be a disruption in one of them, in two of these areas, in all three, in different combinations and different weightings. And that's what we're trying to sort of, in a sense, dissect out. So the great thing, I've alluded to this, is that with the right computational tools, we should be able to study state representation processes or changes in them across species and levels of scale. And again, I think I've already alluded to that if you have a mouse model, you can sort of examine things at sort of much more fine-grained cellular and microcircuit levels. In monkeys, you can move between microcircuit, mesocircuits, and macrocircuits. Of course, in humans, we're focusing on larger scale macrocircuit brain network interactions. And then in humans, we're trying to get much, of course, into then the subjective experiences and the behavior. So what do we discover when we use computational methods as a linking tool? And I've put this illustration from this paper that the whole group of us co-authored on the idea of computational validity, that if you can find a task that is engaging the same computations across species, you're going to be able to sort of now drill down into some and create some of these bridges among the different parts of the elephant. So this is where it's going to get a little bit technical. I'm going to try and focus on highlights because I don't want to lose those of you who don't do this work. And again, from this phase of the talk, I want you just more to be thinking about, oh, this is the general way in which these kinds of methods get applied. And this is a general insight into how we might see more of this happening in the field of psychiatry. So what we've done is we have focused on two different behavioral tasks that can be done across three species and that allow us to probe different aspects of state representation. We chose these two tasks for a number of reasons, but I'm going to outline to you some of the specifics here below. And we have data from the first task, which is called, I'm going to be calling it topics. It's a translational orientation pattern expectancy task. Don't need to worry about that piece of it, just topics. And we've done that task in humans and in macaques. We've tried it in mice. It hasn't worked out so well. We have a new task we're working on that is quite similar. That seems to be working better in the mouse. That's for those of you who do this kind of work. Then we have a translational bandit task. I'll be calling it TBT. And that has been successfully applied across all 3 species. What's really interesting about these 2 tasks is they each probe different cognitive, and I would say, I'm going to show you microcognitive processes that are very relevant to psychosis and that are near and dear to my heart. One is cognitive control. So again, these tasks are not focused on auditory hallucinations or delusions, right? Because we can't study that across species, but we can study state representation functions across species. And one, we're going to be looking at state representation functions when the brain has to understand a context and keep the rules for that context in mind and make appropriate decisions. So this is very relevant to the prefrontal dysfunction that we see in psychosis, the executive control problems, and it's derived from a very well-studied task that people know about in the field. The translational bandit task is relevant to reward system dysfunction. It's a task where the environment is continuously changing its reward values, and you have to be making choices that allow you to kind of gather the most rewards. And so it's very relevant to reward processing, impaired learning, and motivation in psychosis, and it's derived from a set of well-studied probabilistic reversal learning tasks. And so the first one, topics, there's correct or incorrect responses. There's reaction times we measure. You either get it right or you don't. The second one is more, how are you responding? What kind of strategies are you using as reward contingencies in the environment are changing? Are you learning that reward contingencies are changing? Are you profiting from that? So the tasks, very briefly, the topics task is a task in which you get presented different stimuli and then a probe. And when you get a correct sequence, we call it an A Q followed by the X probe, that's a correct response. You respond correct. And if it's any other probe, any other Q followed by any other response probe, that's an incorrect response. And what's done is that it's set up so that the AX pairing happens very frequently. So people get used to A is going to be followed by X, A is followed by X, A is going to be followed by X. And so people get used to responding to that. And you can imagine if an A shows up and it's not followed by an X, there's that, oh, I've got to change. The context is changed. It's not an A and an X, it's an A and a Y. I got to change my response, right? So it's context maintenance. And then, you can see if there's a B probe that the brain is saying, oh, it's a B, I don't need to worry about it. So it's probing these different aspects of, can I maintain context? I've gotten used to a prepotent set of responses, but am I able to sort of estimate what, in a sense, state of the world I'm in on any particular trial? It's an A world, it's a B world, it's followed by the X, it's followed by the Y. Again, I'm happy to drill into details if you want, but the main thing is you have to maintain the task context in order to perform accurately. In the TBT task, what you need to do is you're presented with 3 stimuli and you pick one. Your goal is just to make as many points as you can. And the reward probabilities of each stimulus is walking over time. And that's illustrated there at the bottom for you. So you might have a red, a green, a blue stimulus. Sorry, those got shifted a little bit in the translation. But what you then have is that over time, and the reward probability of each stimulus is walking or changing over time, unbeknownst to the person doing it. And so at some point, you're going to, maybe you've started out doing the red, you're saying red, 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 I'm getting rewarded. And then red starts to not be quite as rewarding. And then you have to figure out, oh, do I stay with red because it was rewarded, or do I start to explore other options? And so some of the things you can pull out of this task, if you're looking at trial by trial behavior, are things like, are people tending to exploit a given color choice because it seems to be giving them rewards, or trying to explore other colors? So it's an explore trade-off. You can look at how quickly they're learning that a given response has a certain reward value. You can look and see how much weighting they're giving to the fact that now the environment's becoming uncertain. So lots of information you can pull from this task. 2 very different tasks, 2 very different sets of neural operations. So different state representation processes, different neural contributors. As I told you, topics, mostly state estimation and stability, TBT, mostly state learning. And what's interesting is they each provide windows into different aspects of some of the kind of major, a sense, sort of hypothesized contributors to psychosis. So in one case, these glutamatergic NMDA receptor cortical neuronal systems, and in the case of TBT, striatal hyperdopaminergia and the effects of dopamine blocking drugs. So again, just to sort of give you a principle, different kinds of models can be applied to what's happening trial by trial as people move through the task. And the main thing I want you to pick up from here is that so one of the models, you're going to probably be hearing about this if you dip into this literature at all, is drift diffusion modeling. This is a way of modeling how the brain is accumulating information when it's presented with a task. And there's actually ways you can model the neural systems, the neural changes that are doing this, which is that neural systems are starting to accumulate evidence that, oh, that was an A. Now I'm going to be expecting that the X is going to come because that happens a lot in this task, right? And so the way this modeling happens is that you look at the reaction time distributions for correct responses, for incorrect responses. From those distributions, you model these parameters, these mathematical parameters. And again, the most important thing here is that as we look at this drift rate parameter, which is how long is it taking for the brain to accumulate the evidence that it knows it's in a certain state, the A state, followed by X, the B state, followed by Y, whatever the combination is, that's the state estimation process. And so there are these parameters that we can derive from this particular kind of modeling. An example of modeling that we can apply to the TBT task is a different kind of mathematical modeling. It's called hidden Markov modeling. And what this allows us to do is infer a state, a latent state that the brain might be in by looking at changes in trial by trial behavior. And so here again, what you see is up at the top, now you see how the probability of each color is changing over time. And what you see across the top is the actual choices that the individual subject is making. And when we apply this form of modeling, you see in the shaded gray areas is the person in exploration or in exploitation. So are they sticking with a choice because it's rewarded, or have they realized the reward probability is changing for this choice, I'm going to move and explore the environment. And so we can model how much time an individual is spending in explore versus exploit as one thing. There are other kinds of models and parameters that can be derived. But this is probing more the state learning piece. You know, is the individual learning that they're in a state, or that they're in a state that's changing? Are they staying stable in a given state in their interpretation of a state, or are they moving along into are they less stable in terms of their state representation processes? So what do we find when we apply these tasks across species? Well, when we do it in healthy mice, monkeys, and humans, we see consistent behavioral patterns that reflect task adaptive, state estimation, stability, and learning processes. Just as an example, up here at the top, I've put for you the pattern that we get from the hidden Markov modeling, which is down at the bottom here on that TBT task in terms of the explore, exploit balance in the monkeys. This is their actual behavioral data. So what we see is a pattern that is where monkeys are alternating between explore and exploit, just like we see mice do, like we see humans do. The other interesting thing about these tools, or these parameters that we pull out, these mathematical numbers that we pull out from these tasks when we look at trial by trial behavior, is that they reveal inter-individual differences. Not all individuals will perform the task the same way, even though they might at the end of the day get the same number of, say, correct responses or the same number of rewards. And so what we saw in the mice experiments, which was really interesting, is that males and females are performing the task differently, even though at the end of the day, this is in mice, they each get the same overall total rewards. So male mice are much more in the explore state. And the female mice have much more stable kind of learning patterns in the state. Not surprising. That was an editorial comment. What we have here then is a Swiss Army knife. So now we can apply a given task to a set of individuals, and in a sense almost have different tools from our Swiss Army knife that allow us to pull out different microcognitive strategies, different ways of approaching the same set of contingencies, say, in the environment, and starting to understand individual differences. That's exciting for psychiatry, right? Because in psychiatry, Anyone who practices clinically knows it's all about the individual differences, right? You can have all of the, wonderful evidence-based RCTs in the world at your fingertips, and it really doesn't matter at the end of the day when you have a patient in front of you who's showing some particular unique features or characteristics. So what we have seen also is that when we look at people of psychosis and then we look at some manipulations in the animals, we see, for example, that our topic state estimation parameters are changing in people of early psychosis just the way they change in macaques who get NMDA receptor disruption, who get ketamine. So that's disrupting this glutamatergic neurotransmission. And here what I'm showing you is that when people have psychosis, the drift rate, their state estimation abilities across actually many of the pairs, the probe Q pairs, but particularly around BX and BY are impaired. What we see here, so here lower is worse on this slide, just in terms of the way that the coefficients were used, higher is worse. And you see the same pattern of changes in these BX and BY state estimation parameters. So this is some computational validity that what we see in patients with psychosis is similar in monkeys who are given disruption of that glutamatergic system. Similarly, we see that the TBT state learning parameters change in humans with early psychosis in a direction that is similar to mice given dopamine antagonism. And we've seen changes, really interesting changes. I'm going to walk you through that in a minute because I think it has implications for the meds we give. So what we see in the humans with early psychosis is that they spend more time in the explore state. And that's because they're leaving the exploit state too soon. And that means even though they're in a state which they should be exploiting because they're getting rewards, something about that is not stable. They're not learning in a stable way that I'm in a rewarding state and should stay here. They're drifting into exploring randomly more their environment. And this has really interesting implications then for what is the brain doing with the information it's taking in from the world. It's not holding on to it. It's not learning from it. It's kind of neuronal responses are kind of shifting around in an unstable way. This is across two baselines. So we see that that's stable. Now, what we see is when mice are given a dopamine antagonist, a blocker, a general blocker, that's the flupentixol, they actually show some slight increase in the exploration. But when they're given a dopamine, let me make sure I get that, yes. And then when they're given the dopamine, so that's similar to what we see in patients, right? This increase in exploration beyond what's normal. So that's the effect of the dopamine blocker. That's the part that's raising the interesting questions about the meds that we give. But when we give them apomorphine, which enhances dopamine transmission, then we see this reduction in the probability of exploring. They're more in a sense, able to be learning from these changing contingencies. Main point I want to make here is that we're seeming to show that we can then see disruptions in psychosis, and those disruptions are paralleling some of these manipulations we're doing in the animal models. So what else do we see? This is, again, a bit more detail maybe than is necessary for a general overview, but I want to just point out a couple things. Our topics computational parameters actually do a better job at distinguishing between those who have psychosis and those who don't than conventional task measures. They're very stable. They show high test-retest reliability. No healthy controls show these disrupted patterns in this topic state estimation kind of parameters, though 15% of healthies show disruptions in this TBT reward processing tasks, suggesting to me that you can have variations in reward processing in ways that may not necessarily contribute directly to risk for psychosis or vulnerabilities, but once you start to have impairments in that state estimation, there's a big problem going on. And then a little bit more about kind of this ability to form and hold problems in forming and holding stable representations. We see that these computational parameters are associated with clinical features. In this case, a topic's drift rate parameter of state estimation correlating with negative symptoms, the probability of maximizing that exploit state I described to you correlating with anhedonia. And we also see associations with neuroimaging features. I think, again, I'm just going to highlight that what you have here is using these parameters during the explore state, which we know patients are staying in more. Even though they're in that state more, we're seeing that they have diminished activation of different aspects of the triple network. And what we have here on the right-hand side, I'm not going to, it's a bit of a kind of a subtle and complex and interesting slide, but basically what this slide is showing, these are EEG measures of how the brain's responding to a rewarded stimulus and how that's influencing what it does next. And what we have here is, again, some of these this disruptions. It's not, what's happening next has no relationship to what it just represented. These computationals are malleable. We see them change in response to cognitive training. I think that's the take-home message here. And if they're closer to the underlying brain information processing, do they help us dissect heterogeneity? And that's basically the punchline of my talk. We can apply these really interesting and complex and fairly novel machine learning analyses, where again, now what we're saying is we've got these computational measures across patients, tell us what groupings there are within them. And we see 3 subgroupings emerge with these different patterns of these parameters. And we get 3 interpretable subprofiles. One that is state representation collapse. They're not doing any part of these processes normally. One in which they're doing stochastic state representation, and one where it's just impaired state stability. And so I had pointed out to you before these different ways state representation can break down. And what we have is subgroups of patients where it's all aspects are breaking down or only several aspects are breaking down. And that seems to be what characterizes these subgroups in very stable ways. This is stable across time. And I'm going to wrap up by just telling you that we've looked at a couple of these subprofiles in more detail. This first subprofile looks like macaques who've been given ketamine very closely. And what we see is that there's some changes as in their cognition, their symptoms, EEG, fMRI findings, which are starting to create interpretable pattern. Subprofile 2, it has some elements consistent with the hyperdopaminergia in mice. And what we see here is also some overlap with some of these BSNP subtypes I was describing to you. And again, some interesting findings in terms of cognition, symptoms, and EEG, fMRI. This is just the start. This is a promissory note. So to conclude with my thoughts, we talked about the elephant in the room, but computational methods might facilitate experiments we can do to kind of put the parts of the elephant together. They could be this linking tool, but also a Swiss Army knife that allows us to dissect out different microcognitive dimensions. And when we just use this linking tool, we're starting to see that there might be different computational subprofiles that are lining up with different neurophysiologic manipulations. And thank you for your attention. We're close to time. I'm happy to take a question or to dialogue individually later, or to take questions over email or Zoom for those of you who do this kind of work. Yeah. Yes. Whoops. Yes. Oh.
Right direct from.
Hi, thank you. That was incredibly compelling case for computational methods. And I have a question about the drift rate findings.
Mm-hmm.
I'm wondering to what extent you're thinking about that as specific to psychosis versus more of a transdiagnostic risk factor for psychopathology.
Yeah, so I think that's a spot on question, and absolutely something like state estimation impairments is probably something you're going to find across. My guess is sort of cognitive phenotypes, more impaired cognitive phenotypes. So then the question becomes, what tips it over to create psychosis? And do you have to have a combination of a brain that is slower at evidence accumulation, plus then say a state of hyperdopaminergia, which certainly many models suspect. We're actually going to try and play around with that experimentally with the animals, is kind of try different levels of kind of creating different states of essentially hypoglutamate functioning, and then adding to that different states of dopamine tone. So who knows? I suspect you're right, is the short version. Yeah.
Talk. Are there developmental effects that you've looked at?
Absolutely. And you know, the piece which I left out, but which I'm very cognizant of, is this notion that it's not like any of these disruptions. It's not like the infant is born with all these disruptions, right? And then just some time passes and then psychosis develops. The brain is born with perhaps some vulnerabilities. Maybe it has a tendency to a slightly slower drift rate. Maybe it has a little bit of a tendency to not assign, reward values and salience very well, right? But then, it passes through infancy, passes through childhood. Maybe this is a child who's just a little bit slower to get things. Now they start to be bullied. Now they start to have some stress responsivity, right? Maybe then there's a mild state of increased inflammatory response due to sort of a chronic stress. Maybe that starts changing how interneurons are functioning. Now you're getting some contribution of, kind of worsened, some kind of a hypoglutamatergic state. Now time passes. Now it's, they're off to the military service, and now it's in boot camp, and now they're super stressed out and they're completely disoriented because they're not at home anymore. And now you have this acute stressor happening, right? So it's definitely a developmental process, and it's challenging both to model that, and it's challenging also to think about how to work with it experimentally. But we've started trying to think about how we might do that within, for example, the mice experiments. Yeah.
Question in the chat, which is, what are the implications of your research on ketamine as a therapeutic agent generally, and also dopamine blockers for psychosis?
Okay, great question. I'm agnostic about ketamine therapeutically, because we've not been using it we've been using it as an experimental manipulation in the monkey model, just to sort of knock out, temporarily knock out glutamatergic functioning. So I can't comment on that as a therapeutic agent. It's not my area of expertise. But dopamine blocking agents, I have to say, and this is so against the lore, not even the lore, it's the orthodoxy. The orthodoxy in psychosis treatment, and particularly in early psychosis treatment, is get people on a antipsychotic that is a dopamine blocker and do it at any cost and give them injectables. And if they're not responding, put them on clozapine, right? I mean, that's the orthodoxy, and that's drilled into us. And there's lots of evidence to suggest that relapse rates, are reduced and so forth if we get people on those dopamine blocking agents. As someone who thinks a lot about cognition and plasticity and motivation, I'm very concerned about that as the orthodoxy. For one thing, we know in experiments with healthy humans, we know that if you give them an antipsychotic medication, they will report anhedonia within a few hours, and you will see a change in their reward valuation task performance. So we're giving agents which are likely contributing to the amotivational state of patients. We're also giving medications, a lot of medications we give to patients generally in psychiatry have anticholinergic effects. And even in meds we don't think of as anticholinergic, and those we know are sort of antiplasticity agents. So we're giving medications that make the brain less able to learn, less able to make use of rewarding information and change behavior due to it. And that's the orthodoxy. I hope that we are going to have a paradigm shift in the next 5 to 10 years. Yes. Thank you.